Shall we start? Okay.
Yes. Yes, sir.
So once again, good morning to one and all, and welcome to the second session. So we have a resource person, Dr. Basappa from Organic Chemistry, University of Mysore. He is a very good researcher working under the guidance of one of the well-known scientist and Professor K.S. Rangappa, former Vice Chancellor of University of Mysore. So presently, Dr. Basappa is working as Associate Professor in Organic Chemistry, University of Mysore, Manas Gangotri, Mysore. He did his post-graduation in Organic Chemistry from University of Mysore in 1999. He obtained his PhD in 2002 and worked on synthesis and characterization of some biologically active heterocyclic compounds. He has completed his PhD degree from Mysore University only. He has been awarded various fellowships. I just quote a few. One is one of the prestigious fellowship, visiting scholar. As visiting scholar, he visited to Mysore University and stayed for a few weeks there and to pursue research work. Then he has been awarded to Dr. D.C. Paute Memorial Visiting Fellowship in 2012 and worked in University of Cambridge, UK. Then later he joined as a postdoctoral fellow, worked in Singapore MIT Research and Technology Institute, so where he has pursued his postdoctoral work. Then also he visited Japan Society for the Promotion of Science. Under this fellowship, he had been to Japan, where Okiodo University, he worked there for some time. Later, he has been awarded CSIR, Senior Research Fellowship, under Medical Sciences in Department of Studies in Chemistry and worked in University of Mysore. Later, he has been awarded again University Grants Commission Project Fellowship for 1 year. So, he has obtained 4 to 8 patents. 8 patents with this young age. He proved himself as one of the excellent scientists in organic chemistry by obtaining 8 patents. And presently, he is guiding the 6 PhD students, and he has published more than 100 research papers. So he has handled worth more than 1 crore rupees projects funded by various institutes like DST, DBT, UGC, etc. With this brief introduction, I welcome you, Dr. Basappa, to this session. And on behalf of all the participants and on my personal behalf, I once again welcome to Basappa. Over to Basappa. Thank you.
Thank you, Basarajappa sir, for a great introduction towards me. So,
thank you, sir.
At first, I would like to understand the participants, how best I can deliver my lecture. So, based on the input and result received by you. Okay, so if you don't mind, so may I know about the audience?
Yeah, surely. You can interact directly with audience, sir, participants.
Yeah.
So as far as my knowledge goes, all the participants are excellently participating with their, with all the sessions. What I have obtained, the feedback from resource persons, all are very enthusiastic. And it is a heterogeneous group, sir. Zoology, biobotany, and biochemistry, biotechnology, and other science department faculty members are joined here. And all are very enthusiastic to learn from the resource persons. This is what I came to know. So this is what the brief information and feedback about the participants. Thank you.
Good. Yeah. So coming back to the introductory platform. So let me open it and sorry. Yeah. So may I go one by one? So can you tell me your area specialization and from which university you are from, so that I can understand? So I believe anybody from chemistry. So
I think no one is from chemistry as such.
Okay.
Biotech,
please tell me about-
biochemistry. Yeah,
Okay.
myself, I am Dr. Anandan from Department of Genetics, University of Madras.
Very good.
So I'm basically a genetics, but I work with natural products as such. So I think that goes well with your topic today. And
Very
I'm happy to be.
Thank you, sir. Thank you. Yeah. Next, please quickly.
Sir,
sir.
Hi.
sir, Niranjan from microbiology, sir.
Sir, namaste. How are you?
Fine, thank you, sir.
Very good. Yeah, good to hear that you are here. Okay.
Yes, sir, yes.
I hope your conference went very well.
Yes, sir.
Yeah. Next.
Hello, sir. Good morning. I'm Kavya. I'm from Central University of Tamil Nadu. I'm from microbiology department.
Okay. So you are a faculty there.
Yes, sir. I'm an assistant professor in Department of Microbiology.
Very good. Area of research?
Sir, I'm working with PGPR on one side, and on the other side, I'm looking at mitochondrial dynamics in yeast.
Okay. Thank you, madam. Thank you. So next.
Sir, hello,
This
sir.
is Aruna, Assistant Professor of Botany, Tyagarajar College, Madurai, sir.
Okay.
Hello, sir.
Yeah, please.
Sir, I'm Dr. Gandhipati from Elahaji Government Arts College for Women, Tirupur, sir.
Okay.
Belong to Bharatiya University.
Bhartiyar. Okay. Bhartiyar. Yeah. Arjunan, sir, he was heading some Arjunan.
Sir,
Department
I'm
head.
from.
I think
I'm
he
from
got
a
retired.
college
Sir,
affiliated
Arjun.
to Bharatiya.
Oh yeah,
University.
I know.
He got
I was
retired.
I was a JSPS fellow. He
Yeah.
visited as a visiting professor that time.
Yeah, he was working on pheromones actually.
Yeah,
basically.
Fairmount.
Yeah.
2007, I think. Yeah.
Yeah.
I met him in Japan. Yeah. He's a good researcher. Yeah. Next, anybody?
Good morning, sir. Myself is Dr. Chaitra from Vijaya College. It is affiliated to Bangalore City University, Bangalore.
Bangalore City University. Okay.
Yeah, I'm my stream is zoology.
Okay, very good. Okay, madam. Thank you. Next.
Sir, good morning, sir. Sir, this is INR from AVVM Puspam College, Tanjavur, sir. Belongs to affiliated to Bharat University. My field of research is plant systematics and ethnopharmacology, sir.
Okay, very good. Okay.
Yeah, thank you, sir.
My name is Lakshmi Khan, working as an assistant professor of biochemistry at University of Agricultural Sciences, Raichur. My research area is to discover novel insecticides from Bacillus thuringiensis strains.
Okay. Biochemistry. Okay.
Yes, sir.
Okay, nice to know, sir. Thank you, sir. Next.
Good morning, sir. My name is Meenakshi Pandey from Biotech Department, GPS Institute of Agriculture Management, Bangalore, sir, recognized by Bangalore University.
Okay.
Sir, this is Paul Darshan from Bishover College, working as an assistant professor in Department of Botany, sir. My research work is based on the hybridization in plants, especially tree species.
Okay, very good. Okay. Thank you, sir. Next.
Hello, sir. Good morning, sir. This is Rupini from GPS Institute of Agriculture Management, recognized by Bangalore University, sir. I'm assistant professor in horticulture and plant protection.
Okay, thank you, madam. Thank you. Next.
Hello, sir.
Good afternoon, sir.
Hello, sir.
Yeah.
I'm Dr.
Please.
Vinutha, sir, from Biochemistry Department, working as assistant professor in Agriculture College, Hanvanmatti, which belongs to University of Agricultural Sciences, Dharwad.
Dharwad. Okay. Yeah. Okay. Yeah. Thank you, madam. Next.
Good afternoon, sir. I am Dr. Bhuvaneshwari from Ayanara Janahima College, Sivagasi. It's affiliated to NTU, sir.
Yes, madam. Thank you, madam. Next.
Good afternoon, sir. I am Dr. K. Jayasilan, Assistant Professor of Zoology, AVVMC Puspam College, Pundi, Bharat University, Tamil Nadu.
Okay. Next. Thank you.
Doctor.
Hello, sir.
Yes, sir. Please go ahead.
जी डॉक्टर धर्मेंद्र कुमार सर, गवर्नमेंट पीजी कॉलेज रायपुर, देहरादून, उत्तराखंड सर.
Uttarakhand. Very good. Okay. Next. Around 22 participants. Around 20 is there.
Dr. Shinaja.
Sir.
Dr. Shinaja, working as assistant professor in zoology, Kerala Verma College, Trishur, belongs to Calicut University, and I'm from Kerala.
and I had done my PhD in spatial cognition fish.
Okay. Calicut, I think Nevin is there, no? Nevin KG
Uh,
from biochemistry.
I'm not in the university, and I'm in a college.
Okay. Thank you, madam. Thank you, madam. Next.
Good morning, sir.
Yeah, good morning. Very good morning. Tell me, madam.
This is Dr. Sayyada Kahkasha from ASM College, Bellary, and my college is affiliated to Karnataka State Women's University, Vijaypura. I'm an assistant professor in the Department of Zoology. My area of research was the aquaculture and hydrobiological studies.
Okay, very good. Yeah, thank you. Next.
Thank you, sir.
I hope everyone got introduced. Okay.
Good morning, sir.
Yeah, good morning.
I am Dr. Kalishwaran. I am Kalishwaran, AVMC Department of Zoology, sir.
Okay. Thank you, sir.
I am working in, especially in the fish immunology and development of the immunostimulant activity using from the plant-based.
Okay. Okay. Thank you, sir. So I hope everyone, yeah. So let me go into the today's topic. So from my presentation, so share screen. Okay. So everyone is seeing my slides. So at first, I would like to bring it to your kind view where we recently our patent got accepted in 13 countries. So US basically from we started from Indian patent and followed by the other European patent PCT filing, and US patent is granted for this novel chemical entities that is targeting for triple factor 3. Okay, so with this slide, so I just wish to tell you that how one we can patent is patent any kind of novel entity or novel resources or novel method or novel structures that are useful to the society or in the healthcare system. So today, so I am going to give a, so yeah, my introduction is given. So as an overview, so this is the slide which will help you to understand the key drug molecule, drug like compounds or drug seed we say. So their target and the naming which we given and their application and patents and R&D stage that we have acquired. So this is the bottleneck or the overall summary of our past or these some of the structures which are really going for clinical trials. So I am emphasizing over here our 20 years of research work, where at first I would like to inform about or give an background of NPB that targets bad phosphorylation. So basically this bad gene, so which is very important for a medicinal chemist, because despite the initial success of therapeutic agents targeting RAS or MAP kinases or PI3K or AKT-mTOR signaling networks in oncology, so where the development of acquired resistance to such a therapeutics represents a significant challenge in successful disease management. So other part was well known, so where AKT pathway or MAP kinase pathway was known, but in our laboratory, I started this research physically. We started this work from 2013 at University of Cambridge when I was a poverty fellow at University of Cambridge and a college that is affiliated with Sydney Sussex College, where Mayal, who is guiding us. So I really thank that time to the Karnataka University, which really sends scholars every year, 2 members, to get knowledge from such a prestigious institute. So at that time, I took this problem, or the medical doctor, MD doctor, from my previous collaboration, or I was a Singapore MIT research associate at National University of Singapore, which got associated with the National University of Health, NUH. So where medical doctor Professor Peter E. Lobby, who interested to develop a new drug molecule for a new or untouched targets, genes that are really responsible other than the traditional targets like AKT inhibitors known, BCL2 inhibitors were known, or drugs were approved by US FDA. So in this case, so BCL2 uh, associated death promoter BAT is a common and core downstream molecule for both the pathways that I have mentioned, like MAP kinase or AKT. Okay, so basically BAD unphosphorylated state, which is BAD that binds to BCL2, which results in the activation of BACs or BAC, okay, and triggers the apoptosis. But our molecule NPB, which we discovered and characterized by preparing many libraries. So this molecule specifically inhibited the bad phosphorylation on serine 99 amino acid residue. Okay, so this we observed by in silico and in vitro and in vivo. Okay, so by this way, the molecule was lead optimized and lead identified, and this molecule was patented finally, the lead structure with a US patent, and now we went with 13 countries patent, and pre-clinical studies almost completed, and we are in clinical trials. Okay, same thing is true with regard to triphyle factor 3, TFF3, a gene. This gene highly expresses in gastrointestinal system, and even in lung cancer. And recently there is a discovery that this gene or a protein, a cytokine that expresses in brain also. Okay, so this is also a kind of good novel discovery from our laboratory, where this molecule named AMPC, I will go into the structure and mechanism and how we arrived at clinical stages. Okay, so in this regard, so we recently have a American team where they are really interested to take this molecule for a clinical trials, hoping this AMPC or its developed optimized structure could be a drug molecule in the future. So coming to the epernase enzyme, so which is a glycoenzyme. So this target, which I started working from my first postdoctoral fellowship, JSPS. So where we took this enzyme because it has a very important role in metastatic tumor cells, irrespective of liver cancer, and in many cancer, this as a in advanced cancer patients. So epernase has a major role. So where we identified in 2010 itself, and in 2007 we put up in Japan patent. So this is under preclinical trials. So where we named it as a DMBO. And again, SIMO is the next version of our one more project that has been well established, and we have published in JBC, Journal of Biological Chemistry, and the US patent was obtained for the molecule named SIMO that interact with the phosphorylation of JAK kinase and inhibited the pathway called well-known pathway JAK-STAT3 pathway in liver and breast cancers, and we have lead optimized and identified and discovered for its application. And presently we are finding, or we are funding with the investors to take up further. So because we are in a academic, we are in a academic setup, so we need really funding to make many innovations. So with this, I would like to give in one or two words where the other target that you know already, many of you are from microbiology or biochemistry, tumor necrosis factor alpha, which is also an important target of in any inflammation, I would say. So not only liver and lung cancers, that model which we have evaluated. So the Indian patent that has been granted for the named compound BIHC. So this discovery is which we have made for the second. This is the only molecule after Roche that has been developed a specific inhibitor for TNF alpha functionalization, and we could able to show how TNF alpha functionalization or dimer disruption could happen by BASC in a in vivo and in vitro model that we have demonstrated. So again, so coming back to my second postdoc at MIT, Singapore MIT Alliance Research Center, we could able to identify a lead structure NIC under the guidance of Professor Suresh Shubra and Ram Sasisekaran, who happened to be the Suresh Shubra sir happened to be the NSF Director America when Barack Obama was the president. So he used to work with him at White House. So Suresh Shubra sir lead this project, and of course along with Professor Ram Sasekaran, he was my guide. So we patented this work. Priority patent has been filed in 2010, where we identified a malarial merocyte surface protein, isoform 12, so that it attacks or it binds to MSP12 by NIC, which was characterized. Of course, you know about malaria. So we need a better drug when compared to the naturally occurring other drugs that are treating now. Okay, so because of pathogenic malarial strain still emerging in the Asian countries. Okay, so in the other, so coming to the NC series P300, so which we did very long back when I was a student under Professor K. Rangappa, and with collaboration from JNCSR. So we started patenting this type of anti-cancer drugs. So that has been approved by PCT, and US patent has been granted for NC type of compound. So quickly, BCB refers with EGFR, which is relatively target tissue is triple negative breast cancer cell. So Indian patent has been obtained University of Mysore, of course in collaboration with or in joint with National University of Singapore. Okay, so SUBR, so this is the unique project where it targets hyaluronidase, a glycoenzyme. So basically my postdoctoral studies purely dealt with the understanding of structure, function, or glycosaminoglycans that are present in disease condition or in including marine models, so which we have seen, and we had a collaboration. So I had a collaboration with Larry Sherman from Oregon University. So we applied for US patent now. So we identified SUBR, its isoform SUBR3 as the lead structure, and we have published this work in GLIA, an internationally a reputed journal. So and then the so this work is further continuation by him, and we are working with that target also in collaboration with sorry. So let me go back to my slides. Okay, here I gave a general overview. So next is let me open. So this is the work which I already gave you hint. If I go back to the original discovery, so sorry, yeah, please. Original discovery I wish to share. So sorry, I am opening multiple files. That time it takes some time. So anytime you can ask me if you have any doubt in relation. So basically you need to understand the drafting of patent and specification, patent office examination, with that is a practice they do, and PCT filing. And I took this slide from internet. I think you can take it also. Basically, which is uploaded by the senior partner of our K&S partners, who really helping us for filing a patent, because we are a scientist. So generally we give the novelty of the drug to them, and we explain them, and then they take the information and file it. So quickly we go with how IPR works, or how we can go, or how we can file a patent. So better you need to understand the company protocol, how, or the government how it works. So quick, so with 5 to 10 minutes I will take it and tell the importance, how best you can make your discovery to be protected in the form of either patent of Indian patent or PCT filing in national phase, and there, and also you can expand your patent ownership to the to US or other countries. So that I will be explaining. So basically the scope of a patent, so you know what is a patent. So what is patentable. So I don't want to speak here much. So overview also you know better. So I believe that these things will be explained by your what to say, by others also. This has been given, I believe. So coming to the steps, like to patent a thing. So the solution or an invention that is required, and its application followed by satisfactory condition could be patentable. Okay, so in terms of IP, Indian law, and what we are practicing, so we need to file a patent. So directly, basically, please note that. So I cannot go directly and file a patent to US. So one thing happened with regard to our patent. So we directly wanted some discovery from National University of Singapore and to file a patent to US patent. So which became impossible, because Indian patent filing is mandatory. Okay. So that is the thing that you should know. So better you need to go and file an Indian patent office. So either through lawyer or through a firm, they do like I mentioned in K&S partners. So likewise, so patent office. So as you know, they are available in Chennai, Delhi, and Mumbai. I don't want to waste time. So this is the office overview. All the examiners. So if you hire or if you pay money to the companies called like K&S partners or some other companies, many companies in India are available. If you provide them, they will be in contact with this controller and controller general and lawyer and review whatever. So the things will go easily. So, okay. So invention, coming to the invention that you know, all are professors. So I don't want to explain this. So basically invention which refers with the fundamental research and improvement on existing art or solving or unsolvable problems of art, or coming to the pharmaceutical industry that I have a knowledge. So if you go with the medicinal chemistry patenting, so basically the novelty matters. Okay. So what novel is? The compound structure is novel. If the structure is already in the public domain, called in terms of publication or in terms of presentation somewhere, or it is in the form of thesis somewhere, so it is impossible to patent. So keep in mind that. So the fundamental research, whatever, it should be applicable basically, as I mentioned earlier, and it should be novel. Okay. And mainly if you are putting a money, and you should understand that it should be competitive to the industry, pharmaceutical industry, if the drug is required. So basically in 2022, so a few drugs, almost below 20 drugs, have been approved by US FDA. In that, a few are antibodies, monoclonal antibodies, and some are gene therapy. So every year, a few drugs are coming up, are with an approval, meaning that still the requirement of a new drugs are, or we can say by organic chemistry, in the discovery, the organic scaffold that we synthesize, about only 5% of the known structure in the nature, I would say. That is the understanding by literature, by our team, stating that still more thing to be discovered, and it is required importantly because the evolution of or mutation of genes in cancer, or if you come with COVID-19, you know how much we are suffering and how badly we require a tablet form of a compound that, or a drug that could really target COVID-19 variant. And again, so you are an expert in the evolution, botany or genetics, you all here. I don't know much about that, but once the evolution of many type of viral genome, or its pathogenicity, or its spreading factor, all is important. So in not only COVID-19, like when H1N1 got originated, outburst was from Europe, but Indians, many Indians, especially a pregnant woman, died. Why? Because the non-availability of a drug, or we are not on time, or prior identification we don't do. We generally treat with an antipyretic drugs and followed by antibiotics. But at the end of when you about to confirm, so I'm speaking about 2010 or 2009, where H1N1 was kind of like Mahamari. So what I wish to tell you, here, when we do fundamental research, a patent, if you wish to require to be patented, so there should be a new thing or new resources that should help in healthcare. Then your patent discovery will go from Indian patent to a global, and then it will be taken up by somebody. Okay. So with this, so how we can think for drafting? Okay. So you have something in your hand. So nowadays you don't want to worry about drafting. So if you have a complete data, if you can give it to some company, they will do it for you. Okay. So with this, we don't want to talk more about it. So before drafting, okay, here the science where you need to understand. I given a kind of more I gave or enrich your knowledge by giving our patent problems, like when we need to conduct a search. So in this case, as per as my knowledge is concerned, so we had a good collaboration with an oncology doctor of NUH. So who given us the problem, which are the genes really that we need to focus, where the industry is working with something that is really some drugs are in the market. So an academic a scientist should think in terms of the problem that is really untouched. So then we could able to even so I don't want to tell that our research is great, but to take you not only in patenting, to publish in PNAs, it is very difficult from India. So hardly in a year, 1 or 2, 3 publications are coming up. Okay. So in that perspective also, so if you make a search, now we have a lot of what to say domain to search many things. But when we started this research in 2000 time, so 20 years back I'm speaking. So we were really in need of collaboration. So now also, so if you identify a target or a theme or a problem, that is very important. Okay. And then make a prior art, and then you see the novelty, and check with the solution obvious. This again very important for a life science professors. So the solution is important. The solution should be a novel or newer finding. Okay. So and again the other parameter, like we should exclude its artificiality. Okay. So as publication ensured, so ensurement means that once you have a patent, so anyhow after 12 months, so I will come back to the timeline and everything. So that should be published. Okay. So with this, so I will take up next. So decide the area of nature of protection. So Paris Convention or PCT or ordinary application. So this slide will give you what you need to do with regard to IP. Okay. So next is the code is not patentable. So I don't want to talk much over it. So I will just skip it. So public domain. So this is also, so I don't want to explain. So publication. So kinds of publication, document, paper or publication should provide unmistakable direction or disclosure of the invention. So that these things, I think I will skip here. So patent specification. So these are all the parameters that you look into, like patent office. Again, the company will take care if you give it to him. So license or assignee, and then if you want to compete, so the patent office will search it and give you your things is not novel. Okay. So let us take you are filing a patent. then they will give query. Okay, your research is published somewhere. So we faced all these things. So the publication was there, or the patent is overlapping with the another. They will give the old patent that have been acquired by somebody. Okay, that time this will be, we need to argue with them through a lawyer. Mean to say, so for this case, we refer to court, and then they will honor. Okay, if our work is purely untouched or novel in terms of its application. Okay, so coming to the technical peers are skilled persons. So basically this refers with the methodology. So again, technical team will evaluate, and we will be providing methodologies and all the relevant information. So next is the competitors. So competitor, generally the industry, pharma industry. If the drug is related, or not only drug, if you wish to make a vaccine of your own kind, or mRNA vaccine or whatever. So I'm not an expert to speak much on it. I'm sorry. So with this, so competitor generally industry. So biotech, either biotech industry, or so many industries will be competitors. So you should be make sure your work is purely competitive also, but also the company is not focusing much on that. So commercial players. So commercial players basically is important. So commercial entity, generally, if our work is really needed to the society or healthcare system, so commercial players, which is also an important parameter. Okay, and coming to the general public. Okay, so again, this is the final. So once you have a patent, so you can drop anywhere. You can drop an Indian patent, trying for PCT or trying for USFDA. Okay, finally, if you have everything, but you can't go to clinical trials, so then again it is ending up there. So if company, you need to approach a company, or you need to approach an investor. Okay, so if the investor is agreed, you can proceed for clinical studies, and then phase 1, phase 2 are coming. So it depends on the documentation also. Again, in the technical peers are skilled persons, so documentation is also a very essential part. So coming to the specifications in this classification, like provisional and complete. So with regard to provisional, I would like to tell you that if you have some new data, you can file a patent as a provisional. Okay, complete means, so if you have complete data, you don't want to be wait. So at that time, complete patent will help you, because provisional patent, you can, if you identify a structure and its application, if it is well enough, you can submit a day 1. Once the data you have, you don't want to worry. Like, okay, I am fine. My finding is very novel and competitive. It is required to the human healthcare or industry. Industry really hunting for this one. So you go for provisional. Okay. So once you go to the provisional, there is a 1 year gap, 1 year timeline. So then complete patent you can file. Okay. So this is the classification or kind. The specification of a patent should be there. So kinds of applications, so like conventional or non-conventional. Again, it depends how you will choose. Okay. So addition of a patent. So that is there. That is the next process. So a provisional specification, again, there is a pros and cons. So when there is an urgency, I already mentioned. So provisional, you should be filed from a day 1. Once you identified a problem, and you have something that is should be protected. So urgency is, if you are in urgent, so go for it. Okay. So then commercial disclosure. Okay. So submission of a thesis. Okay. So you need to disclose it. So that time So provisional will really help you. Okay. So inventors are seniors leaving the company. Okay. So again, so these are all the pros and cons, so that you need to know. I will go further. So this is mandatory documentation I am going to say. So somebody, if you really interested or you need to know, Form 2 should be submitted. Okay. Form 2 along with the inventors, applicants. Okay. Inventors will be there. Applicant will be university or an institute, because they give you the resource. Okay. Nowadays, you can, if you have a data, you can directly write to DBT cell. Okay. So there is every 3 months or every meeting, they online you can submit your data and get it reviewed by them. If really the expert accept, so your patent is through. You don't want to pay anything. So DBT does that. So I know that because we have a DBT funded Northeast project that is running up. Okay. So Form 2 is essential, which really explains or describes the invention. Okay. So the first one is in relation with your agreement with a signature. So where the applicant and inventors are with the title, you will be providing it. And Form 2 describes like your whatever you are writing a paper. Okay. So in this case, so provisional specification. So I already told you it should be completed by 12 months. Complete application you should within 12 months. So definitely, otherwise lapse of provisional specification comes into an account. Why I will tell you is that, so 12 months later, so if it is lapse, so you can't claim, because an Indian IP, if you have an Indian patent, complete patent should be filed after 12 months. If not, your original day, the day one urgently you have identified, so that originality will go. Okay, please note that. So then a complete specification. So in this case, again, Form 2 is required. So I explained that. So patent specifications. So here you need to describe, our description is required. What is the things that you are going to be patented, and claims. What is your claim? This is very important domains. Please understand that. So description is different. Claims are different. Claims really says very specifically what is your patentable entity or patentable subject or patentable product or a solution. Okay. So description discusses the invention. Okay. So that in this description, you will give all the methods and the data or how you approached with the solution. Okay. So claims, again, I will tell you that claims definitely boundary of monopoly. Mean to say that hey, this is my claim. You can't touch this. This is how I would say. Okay. So next, coming, what is a complete specification? Okay. So according to the section 10(4), so here the complete specification is the one you are the sole authority of that invention. Mean to say a best method of performing the invention which is known to the applicant and for which he is entitled to claim protection. Mean to say your scientific entity is protected. Once you have a complete specification in IP, that is fine. Mean to say what? So provisionally you file, but I strongly advise you need to complete that. Then only your claim is, or your invention is protected. A timeline I will speak later. Okay. So coming to the description, so I think I will skip because I will take live examples of my patents and then we go there. So description, novelty, and all we have explained here. So the title, so again it should be concise. That you all know because you are a professor in your field and you know how to put up a paper, and thereby the patent is similar in that case. Okay. So I will skip here. Opening this one, yeah, this one is also not required. So background, sometimes it is required. Okay. So background of invention is required to write a patent. Please note that. But Indian patent doesn't need this one. So I will go here. So prior art. So this is again a prior art in the what is a brief write up of what is known before the invention that comes with the background, and then sets out the problems associated with each of the known art and describe the problem proposed to be solved by the invention. So mean to say that so the prior art is again a collective information where you need to explain. So I already told you India, this is not mandatory. Okay. So coming to the prior art, so like I will skip here. So we go further. So objects of the invention. So again, patenting, which provides purpose of the invention and objects and ancillary objects. So essential aspects and preferred or optional aspects. So these things, please note that. So in the case of statement of invention, so there are claims that you can claim. So again here you should be careful. Like if you are claiming one thing, you can't claim the irrelevant subject again in a claim. So in patenting, you should be known and you should be very specific. The claim should not be unrelated, I would say. Please note that. So in IP, so the claims should be specific, and like if you are claiming for malarial pathogen, our compound is targeting, then you can't say the claim again, it is targeting cancer cellular system also. Mean to say that irrelevant claims leads your patent approval requires, or sometime they will suggest you to make a 2 patent or additional patent. Okay. So detailed description of the invention. So this you know, I don't want to claims. So claims I explained. So okay. So these are all the checklist are do and don't. So I will share these slides. So please go ahead. So this is I don't want. So fundamental drafting again, PCR technique. So this one you please note. So and then drafting, you need to avoid negative example. Yeah, here I would like to take my experience. So basically, when you talk about anti-cancer drugs, so IC50 values, like you take, that is the fundamental, right? Because this is the when you screen a number of compounds and hundreds of compounds, so don't mention more on negativity. Okay, so or don't take the one that is claim should be specific. I already told you, and here it is the example that they are saying that. So like if your compound is breast cancer cell sensitive, mean to say it is in nanomolar, your compound is acting on it in the breast cancer cellular system, but if not in lung, so don't put it as a claim. So this is the word that refers with. Okay, so independently worded claims. So this is again your copyright. So I'm speaking over here. So the invention should be understand, because the lawyer who speak when you go court. So like I explained earlier now. So when you are patented, you are like completely chemistry write up doesn't make any sense, because the people should understand your invention. So mean to say that when you are drafting a patent, so the words or the claims should be understandable to the lawyers and the stakeholders. Mean to say, okay, you will be taking the molecule into the next level, clinical studies level. And the stakeholders or the investors may not be from your field. Definitely. So they may have money, and then how you will understand them. This is very important. So only so your clients should be easy to understand the invention, and easy to search also. Again, some investor nowadays all will search the required material. Okay, so how the things goes, it's like invention, you see. So to tell you my recent literature study in relation with coronavirus drug discovery. So basically, remdesivir was picked up quickly. Why? It was approved in 1960s for Ebola virus. Okay, so then it was tested quickly, and then the molnupiravir and many drugs were a Japanese company gave one more drug in the tablet form. So basically, the industry or the World Health Organization, scientific community, or any scientist think that they want to repurpose a drug. Correct. So I mean to say that if your claim is so clear and if you have achieved a drug, so this could be applicable for the other system also. That is the beauty of invention. Okay. So please note that. So any claim, if it is benefited to the human or the healthcare, so definitely that should be important and easy to license. Again, so coming to the licensing, so your institution should be in a free flow. Mean to say that they should be cooperative with the investors or a company to have a licensing. So if there is no licensing, so many investors will not be interested to about your patent. So easy to establish infringement. This is also very important parameter that you want to note. So coming to the characterization in claims, so I will skip here because once you go through the patent by your invention, so you will understand this one. Okay, checklist. I will just so I will stop this one. The general procedure. Let me go with the science what we have discovered, and so the last slide. Overall examination procedure, like I already explained you. File it either provisional or complete, or request for examination. Definitely, the company will put it up to the respective IP, Indian Patenting or PCT office, and then they will request the examination. So first examination report comes. So 12 months. So within that, the things will be ready. Then please with a response. You means like writing a paper. You will be reviewed the similar way. So there is a response. So reply them, response them, and discuss with them, or hearing at a court, and then acceptance or rejection. It depends on your client. Okay, so this is the overall procedure. So this is how the patent, Indian patent, where we have around 12 Indian patents on hand now. So this will be giving the examples in this form. So your patent, name of the work and the company and where it is granted. Okay. So with this, I will come back to the patents that we have already in our hand. So let me, okay. So this I explained. So this is the Australian patent which we recently acquired. So the patent number is represented over here. You can see, so the name and address of each applicant, like National University of Singapore, University of Mysore, and Bangalore University. So where I was when we started, I already explained this work. So I was an appointee of University of Mysore, Bangalore University, professor from assistant professor from 2007 to 17. So that time this discovery went. So giant applicants. Okay, applicant generally is the institute. Okay, or if you have your own company. So here I believe we are all professors. So so company MD could be the applicant. Okay. So name and address of the patent is, patent number is given, title of the compound, like compounds useful in inhibiting human triple factor 3. You can see here. So what I explained. So the invention, if you search triple factor 3, so this patent definitely comes. So likewise, your title should be catchy. It should be easy to know, and everyone can understand. So likewise, so this is the name of the inventors, which we are all listed over here. So again, term patent of the turn 20 years from 18. So so mean to say another 20 years up to 28, 38. So this is the filled with Singapore National University of Singapore ILO team. Okay, so they filed this one. So this is how the according to the Patent Act. So they will give. Okay, so this is the one, and then here I am. So this one I had explained already. So see now, so this work patent I gave here already, and here the one more thing I want to tell you. Once my publication, my patent is coming to the public domain in the form of paper, so many companies sell this drug. This is also one important observation that you need to know. Once it is coming to the public, but they cannot manufacture and make it for humans. Okay, so because this molecule is still in the preclinical trials and is not authorized, but what the companies, many companies are now using our molecules to research purpose. You can see they say bad phosphorylation, say inhibitor with so and so micromolar, and they will sell 100 mg, get code. So if really the one who really interested in the research, so this is available in the market now as for the research purpose. Okay, so the next is I am going to say this is the discovery how we have made using cheminformatics and chemistry and informatics highly supercomputer facility which I used in 2013 itself. Now we are speaking about artificial intelligence that we used, and we had come up with the repurposing of drugs for coronavirus or whatever they use now. But this has been established in 2010 itself. Okay, so that you need to you need to upgrade with regard to your subject. What is the fundamental research when and how this discovery happened? Okay, that so basically patenting or writing a paper is the same, but here one you are giving a right in publication, you will be giving a right to the publisher. Okay, so that is available for the public domain. But if you are patented and go for the publication, you will be explaining, so I have an equity or I have a patent. Okay, so that so you will not be providing complete rights to the respective publisher. Okay, so this is how we work for a respective project. So in vitro and in vivo, and this one is triple factor 3 a drug which we have patented, and again it is soon it will be taken up for the clinical trials with the American company association. Okay, so these are all the publication. Again, you need to publish a lot of evidences. Okay, so once you made a patent, so patent only doesn't allow or catches by the investors, stakeholders, or companies. They need an evidence in the form of many data. Data is required. Again, the mechanistic pathway is required, and what is non-specific is also required. So if you say about the specificity, okay, how can you rule out this molecule and again the toxicity? Okay, so in the preclinical model, I'm running out of time. Sorry, maybe I can take up another 20 minutes, Basura sir. Hello, up to what time now? We can another 10 minutes now.
Yes,
yeah.
sir.
Yeah, I think yeah, we will try. Okay. What I want to tell you here is that so publishing a good number of or good peer-reviewed journal, because there is a fundamental researcher who works with the same field. So then your work will be evaluated and they will be given more idea and required data to be considered. To tell you honestly, my publications, like I will show here, so with regarding triple factor 3, so the one paper that recently accepted in chemistry paper, with a mechanism after 4 revisions. Okay, so I think this slide, this taskbar are hindering me. Please, sorry. Okay, where I can do here like this. Okay, yeah. One by one. Okay, this is this I saw development. Yeah, this you saw. Okay, this you saw. Yeah, this is the slide I missed to talk about because of the real invention. What went? Sorry, we running out of time now. Let me. Yeah, this is one more point. You please note. This is Japanese patent. How it appears. So though you can't read, but you usually you need to know how the things title of the invention. So again here when this is my second patent in Japan, Japan patent. First patent I shown you about awareness. That time I really had a very difficulty in explaining the English version to Japanese version. So purely the kanji we say. So the words, the sentences should be converted to Japanese thing went. So this work was easier because we paid through the lawyer and company. So they did by themselves. So you can see here application number, filing date, and the registration date and everything. So this is how the Japan Patent Office with their Honko signature. So this is the seal you can see. So this is the seal of a Japanese office, and this is signature. Okay. So with this wait, I will go. I missed something to explain you. This one, no. Yeah, this I explained to you. Okay. Yeah. We will go here and we will go this one. I hope you can see now this data. Yeah. You could see this now. I hope you can see. So this is the paper had appeared in ACS Pharmacology and Translational Science. So here this mechanistic work of a molecule AMPC, we named it monomerization of homodimeric TFF3 by an amino nitrate compound that inhibited the TFF3 dependent cancer cell survival. Okay.
Sir, we are able to see only the file, not the paper.
No, I put it up in the now.
Sir,
No,
sir, very PDF open ಮಾಡಿ sir.
PDF I could see, but
we can't see, sir.
Oh, you can't. Okay, okay. Sorry. Let me take it inside the first news here. Okay, okay. I go here. Never mind. I will send this one to you by I can circulate this. At present, I don't know how to help you to come up here more. New slide. Let me go new slide. Now you can see now.
Yes.
Yes, sir.
Now you can, huh?
Yes, sir.
Yeah. Yes. So this is the one. Why I am bringing this one is that. So any discovery, it is easy to patent. Again, I am emphasizing here. Please note. So you can put it up in some journals. What the discovery you did and you patented it is fine. Your institute is behind you. Oh, make a patent. I will get some ranking point. So they will tell you, okay, you will be doing it. But the same thing, the same amount of work, if you put it into some bigger journal, so it is impossible to publish. Okay. So this is the thing what I want to say here. So when you want to say specifically a compound, here an amino nitride that really targeted triple factor 3, a various parameter came into picture, like how it is interacting, how in vitro it is binding, how in silico you can demonstrate that, and how physically the protein and the ligand is interacting, you show that. So what is the molecular basis of this binding? They will ask you. You can say Western blot, okay, this protein is disappeared, sir. This antibody added. So we saw. No. No one will accept that. So you need to show the mechanism, how the in a molecular level sometime. Okay. So this is how the work should be. So this is accepted in a prestigious translational science journal by our team. So I am one of the main corresponding author. You can see here. So that, and also they published in a special issue, new drug modalities in medicinal chemistry, pharmacology, and translational science. Okay. So such a recognition will be honored if your work is really had a potential. Okay. So likewise, so we have shown all the data that really required, how the triple factor 3 is involved in the proliferation of cancer cell, and then how the molecules affected the proliferation of such a cell entity. Okay. So with this, I think I covered some of the things. So if I had some more time, I could show you the basic discovery that really helping, and HRDC here. And I didn't take my talk actually. That was really having some more data to be talk on this. Okay. Yeah. So this one is this I presented in ISCR meeting in Trivandrum. So glycoenzyme, I was talking here, and go with slides just for a minute. So this is the technique which we used in 2005 itself, robotized technique, how the protein and ligand is binding by using surface plasmon resonance technique. Okay. So from Riken Institute. Okay. So this is the molecules and the binding of with respect to protein, the SPR signal which you have measured, and this is with regard to the in vitro data, how the molecule is inhibited the enzyme catalytical activity, and in cellular system and crystal structure that we grown, and this we have published in Cancer Letter in 12 years back, 13 years back, and how we patented its subsequent structure. Okay. So Epernase again, we had a few more structures. So presently we are also working on this project. See, we see what I want to tell you, there should be a continuity. Like if there is a target of interest, so never mind about the collaboration. Please have collaboration. Write with them and explain your things, good thing, and then make them understand. Because once I completed my work at Sugara laboratory, now he is no more, and I made one more collaboration with the person who really cloned Epernase enzyme. This is what I wanted to tell you. So Epernase enzyme cloned by Israel Ulodowski. So I convinced him, sir, this is the structure which we identified, and this is the new structure which we are developing in my lab. And then he immediately agreed, and we wrote I wrote an email to him, and then we got UGC Israel funding, UGC ISF funding, and then our project again re-initiated. So what I wish to tell you, it's being a professor, every day you teach, na. So research, you take it as a part and try to give more knowledge to your researcher and make them to understand your research problem and take it further. This is my one personal suggestion for you people. Okay. So with this, I think I thank you all for listening my work, and so these are all the data. Okay. Thank you. So any questions open to, you can ask me if I am aware, and if I know, I am very happy to tell you.
Sir, for the session, sir, I have one doubt. So this is Aruna from Tyagaraja College, Madurai. How to know, this is a basic question, how to know whether work has been already published? Because I'm asking for a formulation, herbal formulation, sir. How to know whether that exact thing is already has a patent or something? How to know that, sir?
Yeah, coming to the formulation, madam. So I'm so for anything that is published, that will be in public domain. One thing, okay. What you do, you patent search, you do that, okay. So patent search you can do by either one with the SciFinder that is available at your institute, okay, and later STN is there, that is commercial one. Anyhow, you should hook up with some industry and ask them who really working on that area, and don't disclose your formulation, and request them that the published or patented formulation which are really STN will give you, definitely it will give. Yeah.
Sir, I am Dr. Kalishwaran, sir. Hello, sir.
Tell me.
Sir, I have two questions. One is already I applied for the patenting for my invention of a new instrument regarding the blood collection from the fish. That was applied, and also it is going for examination up to the examination level. So fortunately, I got a job in somewhere. So the letter, and also I am not seeing anything in the websites those time. So that was disclosed now. So it can be easily possible to recover again, or I have to apply for the new one. First question, sir.
Okay. So you left the institute where you patented your equipment application, right?
Yes, sir. Yes, sir.
Okay.
Everything is over. Everything is over. Only the last step, the examination. I didn't see in the website also, because I forgot it for these busy schedules. I didn't see in the websites. So after 10 days only, I see that.
See, examination means, see, what the basic thing that you had applied for patenting, right? Yeah. So examination is completed, and did you get a query by the examiner? You should get a query from the examiner. You should give response, and finally they should agree, and the respective fee should be paid by you, and later the respective authority should approve it and grant a patent. Then only that work is completed. Otherwise you can't claim that you patented. This is what I want to tell you, because once you complete patent or provisional patent you make, one complete patent if you make only 6 months, okay. So provisional patent 12 months. So within that you should be in a position to convince the examiner and say novel and get the patent acceptance letter what I shown to you, like that. Otherwise again otherwise no authority of you to say that you have a patent.
Again I have to apply for that same.
Yes, definitely.
From the beginning.
Yes, sure.
Okay. Yeah. Right, sir.
Good afternoon, sir.
Then another, sir, the invention is there is no further commercialization, but it is new one for the research purposes. Research people can they can utilize it. If I have to pay the money for the patent filing, because for anything, what can do?
I'm sorry, I am not audible. If you don't mind, so I can't hear. The voice was very unstable. Can you repeat again or make sure your
Sir, now it is working.
no, what you do, you write to me. We can discuss that. You write to me. Anyway, my email is given. So you write to me. If possible, I can reply immediately. Hello? Or Rajgopal, your Bhuvaneswari Rajgopal.
Sir,
Yeah.
good afternoon,
Now
sir.
it's fine.
Sir, can any of our finding be pattern after presenting our work in any conference or seminar, sir?
No, if you put up any public domain, this is what I want to tell you. If you put up anything in the public domain, so then your novelty goes wrong. There will be no novelty.
Okay, sir.
It will you take care of that. You will not be putting up your things into public domain.
Okay, sir. Thank you,
At
sir.
least
Thank
for
you.
the structure, you can say applications. You can say no problem. But the originality, like formulation, structure, or the composition, don't say.
Okay, sir. Thank you, sir. Thanks.
Yeah. Please, any more? Yeah, if no questions, so please, I thank you very much for listening. I hope I may not bored you really, because I really covered purely on medicinal chemistry, you all from life sciences, and I hope something would have been benefited. So yeah, have a good day and learn more with this refresher course. I hope these administrators like Baswaraju sir and Malini madam providing you good resource persons lectures. So you will be getting some more knowledge. I believe in this regard. Okay. My talk. So, yeah, Niranjan, tell me.
Yeah, if there are no more questions, I would like to propose a vote of thanks on behalf of this participants of this course. I immensely thank Professor Basappa for his very enlightening talk on patenting. We learned a lot, and I think it will be very useful for us in our further research. Thank you very much, sir. Thank you.
Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.